folks that are just starting out with it, just to give you an idea. It's of course in the field, it's a field test, but there is a written test as well, and we have write in the rain, so if there's weather concerns, we have paper where they'll be able to write, even if we have inclement weather, where we often do. So make sure they're prepared for that. But it's a 60-minute test. Of course, you have 5 team members. You want to make sure that all of your team members are exposed to the concepts within the aquatics section, but we also encourage you to have individual team members or pairs specialize in certain areas, because 60 minutes, of course, is not a whole lot of time. And so it will suit them well if they're able to kind of divide and conquer, and they are, maybe some of them are really studied up on the Clean Water Act or the principles of water. They know that down pat, and others are going to focus in on the chemical testing and analysis there, or the Benthic macroinvertebrates. But again, it's a team competition, so we encourage you to make sure they're all exposed to all of it and they have a understanding of each component, but then adding on an opportunity for them to each specialize in an area maybe that that interests them the most, and that they might be the most interested in pursuing later on, even in a career. Just of course, I think, 4th grade, 5th grade, they start maybe even earlier being introduced to the water cycle, so hopefully they have that down, but make sure you might just want to review that and go over the water cycle. Same with the physical and chemical properties of water, and also some of the frameworks that are here in West Virginia and nationwide, the Clean Water Act and other legal components that affect how we protect water, how we use water, and how it's distributed fairly and protected for natural resources and wildlife as well. So on your tables you have safety glasses, and you each have a Hydrogen pH little blue box, and I've got, just from the pond here, Tim, I've collected some water. So we'll do the pH boxes first. You can go ahead and open those up. Yes, you also have a turbidity tube, and in the back you have a turbidity tube. We may have time for that as well. But they're pretty self-explanatory. If you open it up, you should have, oh good, Tim's brought a beaker, that will help. Yeah. I did not have a small beaker. So you do have a little sample test tube. Yes. And you should also have inside once you have your water that we have from the pond today in your test tube, you'll be adding a drop of the Hydrogen solution. So you have that, and then you'll be comparing it to find your pH of the water that we have today with the little, the color strip that you have inside. So everyone has all the components, right? When you're doing this with your students, you should always have safety goggles. I will let you each fill up your sample tube with a beaker.
I think that sample tube, I'm not sure if I remember the instructions correctly, but it's like a 3/4 fill up. It's not a complete
all the way up.
Fill
Yeah.
up. Mm-hmm. So and unfortunately it doesn't have a line that the marks exactly where the fill line is, so you just kind of have to eyeball it a little bit.
I think you also have the directions are right on the solution, so you can go ahead and look, and you'll see that it says to just add the 1 drop. and then mix, and then compare to your color chart. Help if you hold, you could flip over the poster if you need a white sheet, or your paper if you've got your paper so you've got white behind. So is anyone feeling real confident about a reading? What do you think for the pH? Do you want to share?
7.
I didn't look myself, so I'm going to take your word for it. Yep. So pretty simple, right? And Tim may have brought an alternate pH testing. On the test itself, for time's sake, since we only have, what, an hour or so today, we're only going to do maybe the pH and turbidity. But on the test, it's possible that they will be asked to do other parameters, but it will be a Lamont kit or something same as this, where you will have the instructions. The students will have the instructions that tells them step by step how to go through and conduct the test. And so if you have the opportunity and you would like to practice in the classroom in advance, that's great and encouraged. But if you should not have the opportunity to do that, they should still be able to do it, even if they haven't done it previously, because it will be step-by-step instructions.
A couple of key points about these tests. Typically they are color driven, so you'll see when you add a drop of whatever, you'll see a color change, and that'll give you a range in terms of what the test is telling you. This is a general rule. Determining color is sometimes difficult. So, and it especially, believe it or not, more difficult for males than it is females. So males tend to be slightly colorblind. And when your team is doing this test, it might be a good idea to have a female do this instead of a male. And when you are doing it, like Kylie said, hold it up to something that's white background or whatever, and make sure the light is not behind you necessarily, more in front of you. It'll give you a better color view. Just an FYI. But it's pretty basic. It's a step-by-step. Whatever test you're going to be doing the day of, it's going to be a step-by-step. What's not, but in addition to that, we'll probably ask you on the test some follow-up questions about the test, whether it be pH, alkalinity, dissolved oxygen, whatever it is, what the significance of that test is, what does it necessarily mean that day. There'll be a question like that as well.
But turbidity looks at the amount of sediment in the water, and of course if you have excess sedimentation, that's going to make it very difficult for aquatic life to survive. So with a turbidity tube, you see the 25 milliliter fill line, and we just fill it up. You hold it, let me move a little bit, sorry Davin, over the center, and then you see which one on the chart, which one it corresponds to most closely when you're looking directly overhead and down through the water. So you can see on the chart that some of them are kind of more grayed out than others, and they've become lighter. So that will indicate a greater level of turbidity as you're looking down through. If it's become, you can't see that bottom black and white image very well, then you have a higher level of turbidity. So we do have 3 of those, and so we could go ahead and I'll just fill up maybe the water. Let me see more in my peek. this one. And you may need a little more light than what's offered under here. Or you got that right.
Is it on the circle?
I'm gonna pour this one into our.
That's fine. Now, what do you actually look for here?
You're comparing it, if you hold it over the center, you'll compare it to those outside circles on the outside to see which it's closest to, and you may need to get more out in the sun.
Looking in there in the, like, the dark spots, you're wanting them to figure out which ones is close to the colors. Can they be in between? Because I don't think it's a full 20.
Well, good question, because Tim's argument is no. You should pick the one that is closest to.
Right, because there's no other demarcation in there.
Yeah, that's true.
If you're trying to go in between, it's just kind of guess on your part. And in the science world, we try not to guess. So,
Okay.
you know, make a decision one way or the other.
So would we want to do like the one, even if it's, because I don't think that one's much coloration, so would it be okay to put 0 then?
Yes.
Okay. I didn't know if you wanted to go up or lower. I didn't know if there was.
Yeah, I mean,
Okay.
just get to the what the closest.
Okay.
So we look at this and then we pick the one that it is
Yes.
closest to.
Yes. Turbidity is something that is a combination of sediment and the ability of water to reflect light. So, you know, what happens is when something gets a little muddy or whatever in the stream, light comes in and it bounces around, and that's why the turbidity works. And turbidity is done this way, using a tube, or it's actually a chemical test, where you're comparing it to a very clear water sample or whatever. So it could be either way. But basically, it's the ability for water to reflect light.
Well, and another thing, like Tim was saying, this is kind of a basic test that's very economical. We've got this, you know, type of equipment, and it's something you can do right on the stream side. But if you were out and you were really monitoring the water quality, and this might give you a hint that, oh, there might be some turbidity here, but you might follow up with, you know, either more lab equipment or even bringing it into a lab,
Right.
if this type of test indicated a problem.
But you know, it's just you're going when we grade test, and Kelly will get into habitat and all that kind of stuff. There's often not a specific value. It's a range. You know, these tests are not that accurate in terms of giving you a very specific value. It's often a range. So if your students get close, they're going to get it right.
range.
Right. So it's not something that's you know, if we say 0 and they say 20, okay, close enough. You know, if they say 40 or 60, no.
But maybe we give them the range of 0 to 20.
Yeah. So it wouldn't be a specific value, if that helps.
Why do you feel like the habitat assessment might be important for determining water quality?
There's no life behind water quality.
Right. So that habitat is extremely important for being able to allow life to survive, of course. So embeddedness is just looking at the amount of sediment that is surrounding the stones, the cobble, and the bottom of a stream. So a lot of aquatic life, whether it's fish laying eggs or whether it's the benthic macroinvertebrates finding places to live, they really depend on there being less sediment and more of that rocky cobble where they have room for laying eggs or living, and there's water flowing through and they're able to get their oxygen. So embeddedness looks at the amount of sediment that's surrounding the cobble. And so a higher degree of embeddedness is, you know, that's a bad thing. You want less embeddedness. And you have on the habitat data sheet, it gives you, you know, the 4 different levels, whether starting 1 through 5 being 4, and then suboptimal, or marginal, excuse me, suboptimal, and then optimal. And so the students will need to be looking at the stream bed and determining which it fits under, and it's telling you for optimal, that means that less than 10% of the spaces between the gravel and the cobble is surrounded with sediment. And on the other end of the spectrum, fine sediment, that silt or mud, could be surrounding greater than 60% of the cobble at the bottom of the stream for what you might find in a poor stream, rated poor for embeddedness at any rate. You're looking in the riffles. You're not looking like at the stream edges, so that's an important thing for them to know is they're looking within the riffles in the stream. On the data sheet above embeddedness and next in your handout, it talks a little bit about sediment deposition. And that might sound like it's the same thing as embeddedness, but it's not. So it is related, of course, to sediment again. But this time, instead of looking around what's surrounding the individual cobbles, you'll want your students to be looking at the stream at a broader perspective so they can look to see if there's evidence of sediment building up and even forming like islands within the stream. And you will often find those in curves or anywhere the water's been forced to slow down in a pool, and it might build up a sediment island even. So you're looking for those larger features that are evidence of sediment in that case. And again, it's got the grading rubric where they can determine for the entire reach, and like Tim mentioned, that may be up to 100 meters, that they're trying to give it that score. Moving on, they'll also be asked to look at the bank stability and erosion, so looking at the banks on both sides now, and they'll be giving a score. You'll notice instead of the scoring rubric going from 1 to 20, instead of that, like it is for sediment deposition and embeddedness, for the bank stability and the riparian buffer width, they're looking at each bank, so they're giving a score for each side of the stream or river. And the sides are determined by looking downstream, so if you're looking downstream, then this would be your right, and this would be your left. So again, the bank stability, looking at if there's evidence of erosion, giving a score for that. And then the riparian buffer, of course, is the riparian zone leading up to the stream on either bank, and looking, is there a good mix of vegetation? Are there large trees as well as shrubs and even the herbaceous, the smaller plants? You want to have a diversity of plant life ideally. You want to make sure there's good cover. A lot of mountain streams and these aquatic life is really dependent on cold water, so that riparian buffer is not only holding the stream bank in place and preventing erosion and capturing sediment before it gets in the stream, but it's also providing shade, so it's keeping the water temperature lower, which is another component that's really important for a lot of these aquatic species.
When the students are doing this, it doesn't hurt to add comments. Like if they see something a little unusual or whatever, because when we make a decision with a rubric type of situation like that, justification is important. So if I see something one way and you see something another way, convince me. Yeah. Convince me that, okay, I saw, maybe there's something I missed. Maybe there's something you missed. Ladies and gentlemen, this is a pretty critical part of the aquatics test, primarily because it's worth a lot of points and it takes a long time. Usually you need at least a minimum of 2 people working on this. One person's not a good idea. I say 2 people because it could be a little bit of a safety issue from time to time. You're walking along a stream, there's water, there's rocks, there's things that can happen. So having another person around is a good thing to have. But you'll be using a habitat data sheet that probably is going to look exactly like the one available at test day. So you're getting sort of a preview of what the test habitat sheet's going to look like. And unfortunately, we do not have a really good reach to practice on, and we also don't have a tape measure. When we get on competition day, the stream reach is 100 meters long, and you'll be shown the beginning and the ending point, and basically you evaluate a stream reach that's 100 meters in length.
There are multiple habitat conditions that you're going to evaluate, and it's not as hard as it sounds or as difficult as it could be, because if you just follow the data sheet and read what it says and look at the stream, you're going to be okay. Basically, you're trying to get a score as close to myself or Callie as you can, but it doesn't have to be exact. We're a little bit lenient one way or the other. What we do try to emphasize is you need to, when you evaluate the condition of the stream, you're going to come up with an overall score, which you're going to write on your data sheet, and you're going to have an integrity rating, like good, fair, poor, that kind of thing. And as long as your overall score and integrity rating is in the same category as ours, you're good. Okay? So it doesn't have to be exact, it just has to be pretty close. There's also some pointers, some keys on the data sheet itself that helps you evaluate these conditions. So again, read the instructions. That's really important with all these tests. When we get to the chemistry part, when we get to this part or that part, read the instructions, take your time, and we're also there to help you practice and help you answer questions and things like that. Okay? So what we're going to do is we're going to practice a little bit with the data sheet, evaluate a couple of conditions as a team, and then you can talk amongst yourselves a little bit, and then we can talk to us, and you can get our opinions on what you're seeing, and just see how this goes, right? Mm-hmm. Okay, ready? Yes. Okay, so the stream is right behind you.
You're thinking sediment deposition, what is that? And you wanted some hints on that. So you've got that on the back that says hint. Sediment deposition may cause the formation of islands, point bars. So those are the types of things, the islands and point bars are the kinds of things that you would be looking at for sediment deposition. And they give you a little more information on that there. So you see for each one, you're going to give it a score of up to 20. So 1 to 20 for sediment deposition and embeddedness. For the bank vegetative protection, bank stability, and the riparian buffer width, you're evaluating on each bank. So therefore, you're giving a score of 1 to 10 for each side. When you're looking at the side of the stream, any of you like to go kayaking or boating, do you know how to determine the right or left side of a stream? So you're always looking downstream. So in this particular case, our stream's going this way, so that would be the right bank, looking downstream, right? The one across from us, and this, the one we're standing on, is the left bank. Does that make sense? So that's how you determine that. There's a box for each score that you're giving, and then you're totaling those up at the bottom. And like Tim was mentioning, whatever your total score will let you know whether your stream for the habitat assessment is within optimal, a score greater than 80, suboptimal between 60 and 80, marginal 40 to 59, or less than 40 being poor. So those are those categories. And again, I'll just reiterate what Tim was saying. If you're thinking, well, I don't know, for sediment deposition, it could be a 17 or maybe it's an 18. Don't get too stressed about just the single numbers, because we do provide a range that if you're within that range, we're going to agree with you. And you'll get the points for that question or that part of it. Especially if you're you'll notice how each category is aligned with that optimal, suboptimal, marginal, or poor. You just kind of follow those up. So if you're within that range, you think, okay, this fits that category of optimal, that's going to be good enough for our purposes. So again, we would have a 100-meter reach, and it will be reeled out with a tape reel, so it'll be easy for you to see. There'll be flags at the start and at the bottom of your reach, so you know where you're evaluating. I had a workshop on Friday. That's my fault. We don't have a reel today because it broke at my workshop on Friday, so I apologize for that. But sediment deposition, again, you're looking to see if you see any islands. This is a very small stream, so kind of envisioning now that when you're doing the test, we'll be at least a slightly larger stream than what we have here today. So you're going to see if you see any islands or sandbars, that kind of thing, where sediment is collecting. can imagine if there had been a really, you know, a lot of rain or even a flood. These are the types of things that will cause when that sediment drops out, that create these formations. Does anyone want to guess why that could be a problem if you've got too much sediment? Every river system is going to have some amount of sediment, but if there's too much sediment, does anyone want to throw out why that could be an issue? It causes blockages. And also thinking about the life forms and the aquatic ecology. Yes, go ahead.
There won't be enough room for certain animals to hide.
Right. So a lot of our invertebrate communities, especially like that rocky bottom stream bed where they have room, you know, to have oxygen and underneath the rocks to live for their habitat as well. So that plays into embeddedness as well. Our next category that you'll be evaluating on. Does everyone know what a riffle even is? So starting with that, that's the area when you see in a stream. And today, again, I don't know how many we're going to have, but the water's a little lower than when I came out here to check out the stream before. But any location where if you have, especially in higher gradient streams where it's moving from higher elevation to lower, it's moving faster downstream, it's coming over the cobble, the rocks in the bottom of the stream, and you get those air bubbles popping up. It's almost like a little tiny rapid is another way to think of it. You know, those little riffles in the stream. So embeddedness, you want to find one of those riffles. That's where you're going to evaluate it. So within your stream reach, you're not just looking anywhere for embeddedness. You're going to be looking specifically at in a riffle where it's coming down over those rocks. You've got the air bubbles coming up. And then you see within those categories, poor, marginal, suboptimal, and optimal, they're showing you with those little drawings how much sediment surrounds the rocks, and you're trying to see which one does it match closely with. So embeddedness is looking at how much fine sediment is surrounding those larger rock cobbles in the bottom of the stream. And again, the reason for the importance of that is if there's too much sediment, it's filling up around the rocks. That's taking away habitat for our macroinvertebrates and fish species, and it's cutting down on the water flow underneath and around the rocks that's bringing in oxygen. So if you have less than 10% of the spaces around the rocks, you're basically just seeing that rocky cobble in the bottom of the stream. That's evident of a really high quality stream, and that's going to get your higher scores closer to a 20. If you're seeing mostly sand or silt, you can hardly make out the rocks in the bottom of the stream bed, that's going to be a poor quality stream as far as embeddedness goes, and not providing enough habitat. And then the last three, the bank vegetative protection, the bank stability, and the riparian buffer width, are somewhat related as well. They're going to be protecting the habitat within the stream. They're keeping too much sediment from flowing into the stream channel. So with bank vegetative protection, you're looking at the banks, of course, and just seeing how much of them are covered with your grasses and shrubs and trees. And it gives you the rubric there again. So you'll be walking the reach and deciding which one it fits within. Greater than 90% of the banks covered, that's your optimal range here. And again, you're giving a score for the right side and the left side. Bank stability, you're looking at if there is evidence of where the sediment is exposed. So if you had, in this case, we do have a nice vegetative cover, especially right directly next to the stream. But you can imagine in some places, whether if there was a lot of development or a road coming through or agricultural fields that were coming right down to the stream and they didn't have that buffer, you might see more evidence of erosion and sediment getting into the stream. And then the riparian buffer width, you're looking at how wide is the buffer on each side. So again, for a natural buffer, looking at not just whether there's vegetation at all, but is there that mix of natural vegetation where you have the small plants, the herbaceous layer, as well as the shrub layer, and then a mix of trees as well would be ideal. For the optimal range of that, you want that natural mixed buffer out to 60 feet from each side of your stream. So if it's not the full 60 feet, then you're probably going to be docking a few points. And again, using that rubric. So now it's going to be your turn to kind of work your way through. Start up at the top at the sediment deposition. And just between where I'm standing and where the next group is, so we're probably not a full 100 meters here, but you could pace it off if you like to see. But just get a little practice seeing how you would score each one of these parameters. And then we'll come back and discuss our scores and see if we can come up with a consensus and see where we're at on each. So this is just a little time for you to practice on these.
And we saw a wide range of sediment, but we took your tip and we went down and overturned the rocks and saw how the sediment had stained the rocks, and we came up with this is embeddedness,
Yes,
right?
ma'am.
We came up with a score of 7 for this particular
Okay.
stream.
And 7 it falls in what category?
In the
Fine sediment surrounds 30 to 60% of the spaces between the gravel,
Oh.
cobble, and boulders. That one, right?
Yeah, so if you look at the sheet, yeah,
Oh.
there's categories,
Hey, look at
optimal,
that.
suboptimal,
marginal.
Good, that's what I wanted to know. Good. Anybody else do embeddedness? Have any, yes, what do you guys think?
We got 6.
So 6 and 7, that's pretty
We
close.
also
Got a
noticed
different one.
Yeah,
that where the water sits, sediment also sits. So
right. What the tip that she talked about, what I showed them is sometimes if a stream is really embedded, there'll actually be a line on the rock, so they'll be stained where you could see sediment. So you could actually pick up a rock and figure out the percentage just by looking at where that line is. So that's a tip. You know, that only really works on low water conditions, so we don't know what we're going to have, obviously, but that's one thing. So 6 and 7, that's right on it. That's good. It's really good. Okay, let's...
mean, Tim.
We're going to Canada.
Definitely not in my team's body. Did any of you work on it better than individually and want to share your score? These guys looked at it, but not a score. But you don't.
Which one are we looking at?
They're still talking about it.
Probably for like that big tree.
So they were as low as 4. What about anyone else on our end?
We also got a 7.
A 7, yeah.
Okay, so really pretty close across the board. That's excellent. Did anybody do, I know some people did bank stability and buffer width, and did anybody do sediment deposition? That's probably the most difficult condition to evaluate.
No, we didn't.
Anybody tackle that one?
No, they didn't. The overachievers right here.
Okay, what do you think?
I would have called it like a 4. You've got heavy sediment throughout the entire stream. There's just a couple places where you could see where there was a nice area without like a huge amount of sediment on the sides of the stream. From bank, like as a bank getting cut through. That's what I
Okay,
called it.
and when you looked at that, what did you look at?
So when the stream's coming through, if you see an area where there's like almost like a sand bank that's coming through and the water like gets, what am I thinking of?
They're called point bars?
Diverted,
Is that,
yeah,
that's what
yeah,
you're looking for?
yeah.
Okay, yeah, good. Okay, well, I'm not disagreeing with you. I didn't look at this stream, but yeah, you just look for, when you're talking about deposition, you look for areas of depositional, you look for depositional environments. But when you're looking for depositional environments, you also need to think, is this a depositional environment or is it not? And if it's not, if it's like a real fast-moving area, straight, you know, and there's sediment there, that's a problem. If it's a meander and you have a bar on the one side, that's a depositional environment where it should be deposited. So you need to think about those things in terms of that. Okay, good. What about bank stability? Who wants to talk about that? Okay.
we saw that the right side seemed relatively without erosion.
Okay.
There was a lot of vegetation, like all the way down, which is a good indicator. But on the left side, there were a lot of exposed roots that showed that there was healing over time
Okay.
to keep everything in place. But there was evidence of erosion in that.
Good. So why do you think that is? Because, you know, if you look at this kind of environment, this is a very controlled environment, right? You've got the pond up here, and the discharge from the pond really controls the water level in the stream. So it doesn't make a lot of sense that one side would be different than the other, right? Why? Why do you think that is? I'm just throwing it out there. You don't have to know. What do you think?
Different soil on one side.
Maybe. My thing is probably the way this landscape is manicured. So you've got a lot more mowing and a lot more manicure on your left-hand side than you do on your right-hand side, and the runoff is a little bit more powerful. Plus you've got basketball courts and things like that on this side, so that's probably one of the reasons.
Yeah.
Okay. What else? What about buffer width? What do you think?
We said that the left side was a 2 because you've not even got, I stepped it off and you don't even have 20 feet from where we were to the other side. And then on the other side, I called it a 7 because you can see the disturbance. There's barely where we were. There was like no shrub layer, and there's an open spot in the tree canopy. And so we called that a 7. Suboptimal.
Okay, not perfect, but no, not bad, right? On the right-hand side. Okay. What about you guys? Anybody evaluate riparian buffers on the upper part? Same score. Okay.
We had an 8 to the right.
Okay, that's within the same category, and that's what you're really shooting for here. You're not going to get exact. Well, you might. I don't know, but it's most likely not going to get the same score that somebody that has experience, because what happens at competition day, we select the reach. We go up and evaluate the reach, usually a day or so in advance, compare notes, and look at it, maybe go and evaluate it again, and maybe again, and come up with a score. In the past, it was just an overall score and overall integrity rating. But here in the last probably 5 or 6 years, we actually take the time to look at each condition and see if you get it right. So you're going to get some points for having the right evaluation for embeddedness and deposition. Ideally, you want everything right. That's going to get you a better score. But you're also going to get credit for your work. Another thing that you might want to consider, which we didn't talk about at all, is draw the stream ridge.
What is it?
Oh, you did?
What did you say? No.
Oh, okay.
Oh, draw.
Yeah.
Got it.
Okay. And you don't have to be an artist, but if you can justify your evaluation and you draw it and show where it is and maybe provide some notes, any additional information to justify your argument is going to give you a better chance at a better score. If you just give us a sheet of paper, because this data sheet is going to be separate, and it's just circle a few things and you make a few marks, you might have guessed right, but in our world, a guess is not good enough. It's got to be justifiable. So you need to show your work. It's just like doing a math problem. Show your work. That's really important.
I have a question.
Yeah.
The scale bar. So for like embeddedness, it's less than 10%. So if you divide the 5 out, like if you wanted to be more accurate, that represents a 2% change. But then if you go to the 10 to 30%, that's 20%. So do you divide it out to get the percent change for each? Because it's not a true, like, how do you know whether to rank it as a 10 or a 6 or an 8? Is that an approximation once you determine it's going to be in this range?
It is an approximation.
Okay.
Yes.
Okay.
But, it's also about it's a combination of things. I mean, you're doing the math, that's good, but if it fits the description, that's kind of where you go.
Okay.
Start there and then do the math.
Just, okay.
Yeah.
Okay. So how did you overall rate the stream?
Well, that's a good question. I've not looked at this yet, but we've heard all kinds of things. We've had some marginals, we've had some suboptimals, we had some 1 or 2 poors. So what do you think?
Marginal.
Everybody agree?
Yeah.
Well, I would agree with you. So we're all marginal. So you guys are set. You can do this, right? I really strongly encourage you to, if you get the opportunity, to practice. use the same data sheet. We can provide as many as you need, or you can go and download the form or whatever. Practice, practice, practice. Look at a few different stream gradients, stream types. Nothing too large. This doesn't really work very well on a river, for example. This is for streams. And we're talking about first and second and maybe third order streams. And if you don't know what that means, that's part of this aquatics test too. So just practice. And it's better if you can find someone with a little bit of experience, maybe to go out with you. And in your area, who knows, there might be somebody, aquatics professional, or you can call us and we can figure it out and help find somebody that can practice with you. Okay?
Watershed associations,
Right.
conservation districts often have those types of personnel that
Right.
might be able to help as long as they're not on the committee. One caveat to what Tim's saying is that sometimes we get asked, and especially right before a test, we can't go out and help because that might be seen as, especially if it's really close to the test, giving too much help to a particular team. So not someone on our committee, but certainly there's other people out there who can help you find them if you don't have that person already in your contact list. We only have about 10 minutes left, so if you wanted to quickly be able to, we have out turbidity tubes, pH test kits, and I have within these, there's also dissolved oxygen and alkalinity, maybe an iron in there. If you want to just spend a few minutes practicing one of these test kits, you can. And just a reminder, in general, when you're collecting your samples, you're going to be standing downstream from where you're taking your sample, or if you're actually in the stream. In this case, you're probably going to be, it's such a small stream, you're going to be able to get in without actually having to get in, and I know you're probably not prepared footwear-wise to get in. But if we were getting into the stream, you would stand downstream, you would reach with your sample, whatever you're collecting your sample bottle with, upstream, do 3 rinses, and then collect your sample, making sure that you're in a run as well, where the water's moving quickly. You don't want to be over in an eddy or in stagnant water. You want to get your water sample where the water's moving quickly. Went ahead and walked around with just a few images of different benthic macroinvertebrates. So benthic, they live on the bottom. Macro, we can see them with our naked eye. And invertebrates, of course, no backbone. And so they are a good measure, good indicators of water quality, because some of them are more tolerant of pollution, while others are very intolerant of pollution. They're very sensitive to pollution. Therefore, if you're only seeing the tolerant species, that could be an indication you have a water quality problem. So you want to see a lot of biodiversity. You want to see a nice mix. You not only want to find abundance, but you want to find evidence of species richness and a lot of diversity in what you're finding. And hopefully if you're in a mountain stream in West Virginia that has the conditions with the riffles, you're hopefully finding a lot of mayflies, stoneflies, and caddisflies. So I've given you each a strip, and just because of time, that was like in the tiniest little nutshell. But I'd like you to use your field guide, which looks like this, and just see if you can practice identifying what you have on your strip. And when you feel ready, you want to see if you've got the right one. I've got on the end of the table here, hopefully the correct answers. Tim will check my work. And you can come up and see how you did, or you can ask us. We can come around. You can also certainly keep looking at the delineation. This is all for you to take home. But this walks you through how to do a watershed delineation, which is just looking at a topographic map of a watershed and determining the boundaries of the watershed based on the topographic features. So it just walks you through step by step how you would do that, and following, they need to identify where the mouth is. So first need to know which watershed you're going to be delineating, finding the mouth, and then following the topographic features as described and going ahead and mapping the boundaries. And when we did it virtually, and I think you have an example within your folders, they were just selecting which one would be the correct delineation. However, when the test is in person, they will be drawing it, right?
Right.
And so for the virtual test, it was too complicated to try to figure out how they might be able to draw it, you know, and then get it submitted. And we decided to, for simplification, to have it be a multiple choice question. But just know that we're certainly hoping this is going to be in person this year, and so it's likely they're going to need to draw it themselves. And so I've given you, this is Indian Run up at Cacapen, and the examples, one is correct, the other three are not. And so if you wanted to take a look at those, and you could use the directions that are found if you need a little help in the larger booklet. And then when you're back with your students or at home, and I encourage you to go through the whole process step by step, but today, with this being a 20-page long process, we won't do that today.
This is a difficult process.
Yes, and I just saw the time.
And it's a difficult process for the students too. So just kind of if you want to focus on something with the students, and just a basic overview of topographic maps. You know what a contour line tells you, how to read a topographic map. You know what a blue line on a topographic map means. You know what, if you're looking at a topographic map, how can you know if you're looking at the top of a hill, what is a saddle, those kind of things. Because it's, you know, again, this doesn't have to be perfect, but you know, we want them to get as close as we can. And being able to follow contour lines along the boundaries of a watershed is pretty important. I mean, nowadays we have GIS and we have a lot of computer models that do it for us. It's easy, but if we don't understand how we got here and how we determine this, the basic part of it, then we don't really know much about it. And when you're following contour lines, you know, water flows, when you're looking at a mountainside, water flows perpendicular, so it flows down the mountainside. And when you look at this is just kind of tracing the stream.
Right.
It's not following any contour lines or anything, it's just
the outline.
just the outline of the stream.
And this would be the one that follows
This
the
is the one
contour
that
line.
follows the contour line. So
Okay.
basically what you do is once you are at your mouth.
Because if we're looking at a watershed, it's not just the streams, it's also the runoff.
Right.
And this doesn't follow the runoff.
No, it does not.
Okay. I
So
understand
basically,
that.
and it's, again, it's hard to see, but you want to draw your line, you start here, and it's perpendicular to flow, and you go to the highest point, which is about right here,
Yeah.
and then you can make another mark. So basically, you're making marks all the way around your stream, or along the highest point, and then you're connecting the dots.
I get it now.
So this one is the correct one. This one is not, because it's not perpendicular flow. You're going way off over here, and you're actually in another watershed, completely in another watershed. So this one is
And
most
this
correct.
one takes half of it all.
Yeah, this.
And this one does not follow the
Right.
stream runoff from the mountain.
Right.
Okay, so I get it now.
I'm starting.
If you have members of your team that are really interested in, you know, the chemistry and looking at that and doing the kits, you could have certain members working on that, while others might be really great at their benthic macroinvertebrates and identification of those and knowing their tolerance levels and how that relates to stream ecology, and they could be focusing on that. You might have others that are focusing on, you know, the policies and regulation that goes along with water. So we're just encouraging you, because it is a broad test that covers a lot of subjects, and you only have an hour. it's really going to benefit you if you can kind of divide and conquer and have different students working on different parts of the test while you're there. I encourage you to look at the USGS has a really nice interactive website on the water cycle where you can, hover over and click on the different components of that. Go to the advanced level. There's a beginner, intermediate, and advanced level. So go to the advanced level of that and take a look at that. That could be something that you'll see on the test. I'll also be sharing on the Envirothon's website under our Aquatics resources. There are some nice videos that can help you preparing for learning the physical and chemical properties of water. That's something that you should be familiar with. We also have the basics of the Clean Water Act. Of course, that would be something that could be relevant that you might find on the test. And we also have additional videos that I found this year. We have within your folder, you have information on watershed delineation and how, step-by-step, how to do that. But there are also some nice, if you thought a video might be more helpful for your team or your students, there are some that I found online. We'll be adding those to the website. Those are new for this year on how to do a watershed delineation. So that's definitely a concept that if you're not already familiar with, that you should take a look at and get some practice in with your team on doing those watershed delineations, as well as, land use impacts and how they potentially can affect the water quality of a stream.